Did you know that the Zaire Ebola virus, which plagued West Africa and caused over 11,000 deaths, may not be the causative agent for Ebola? Well, at least according to Koch's postulates.
In 1881, German physician Robert Koch published a report that advocated the need for isolating disease-causing organisms in pure culture and explained how to obtain them. 3 years later, while establishing the etiology of anthrax and tuberculosis, Koch and fellow physician Frederick Loeffler developed a list of criteria to help scientists isolate and identify causative agents of various diseases. Koch eventually refined these guidelines into what came to be known as Koch's postulates.
The 4 postulates were as follows. The microbe must be present in all organisms suffering from the disease and should not be found in any healthy organisms. Once identified as a potential causative agent, it should be isolated from a diseased organism and grown in pure culture. The cultured microbe should cause disease when reintroduced into a healthy organism. Lastly, the pathogen is re-isolated from the diseased experimental organism and identified as being identical to the original causative agent.
Let's now talk about why Zaire Ebola might not be responsible for the Ebola outbreak. Koch stated that any infectious particle must be able to go through the sequence of isolating the particle from an organism, growing in culture, and showing similar symptoms when inserted back to a carrier. But Ebola does not follow these principles. There was one key error in the process of determining whether Ebola virus was the causative factor for the symptoms that the carriers of the virus emitted. When testing for the effects of Ebola virus, samples of the virus were extracted and cultured in vero cells. They were then reintroduced into guinea pigs. However, the new strains of Ebola virus showed significantly lower virulence than the original strains. This means that the symptoms present in the guinea pigs did not match those of the original carrier and does not satisfy the third postulate. Therefore, we can confirm that the Zaire Ebola virus is not the causative agent. Well, not exactly. Similar phenomena have occurred in numerous other infectious particles. For example, the measles virus lost pathogenicity when reintroduced into a host organism.
What causes these changes in virulence between stages 2 and 3 of Koch's postulates? There are 3 main factors that can induce change in cultures. Temperature selectivity causes certain strains to be killed in order to best adapt to the temperature of a culture. Receptor switching can cause mutations that alter receptor usage. Natural selection can allow for mutations to develop within cultured cells, creating new strains that lose virulence.
So then, what exactly happened between stages 2 and 3 that caused Ebola to lose virulence? During culturing in the second stage, an addition of adenosine nucleotides in a viral gene caused the virus to become attenuated. Measles encountered a similar problem where the pathogen lost the ability to inhibit interferon signaling, an important immune response. This caused the pathogen to be attacked by the immune system and lose their ability to induce disease.
Koch designed a well-made procedure that would serve as the standard of determining causative agents for infectious disease. However, the nature of genetic errors has made these postulates incorrect in several instances. One key factor to work on would be limiting viral adaptation to maintain as much of the integrity of the original virus. This can be achieved by more accurately replicating the conditions of the host in the cultures. As a result, more realistic interactions between a host cell and pathogen will occur. So as we work toward better control over culturing pathogens, we may be able to argue whether or not Ebola virus meets the conditions necessary to be a causative agent. More importantly, we can strive toward creating ideal culturing environments to more accurately assess and determine the causative agents of a disease and overall increase the quality of healthcare around the world.